So now I'm going to give you an overview of bacteria. A prokaryote is a single-celled organism that lacks a nucleus and membrane-bound organelles. Bacteria and archaea are prokaryotes belonging to distinct domains. DNA sequences and chemical composition distinguish these domains. The first cells were likely prokaryotic. They have thrived for billions of years. This is a really good table that you should take some time to actually read through. So features that are unique to bacteria, unique to archaea, and then that are common between both of them. So talking about some of the major parts of a prokaryotic cell. First of all, the nucleoid. The nucleoid is the region that contains the DNA. Instead of having a nucleus, they just have a pile of DNA in the corner of their cell. And that region where the DNA happens to be is called the nucleoid. Plasmids are small circular pieces of DNA that contain non-essential genes. Plasmids often contain extra and beneficial genes, but those genes are not essential for the survival of the organism. Ribosomes make proteins. A pilus is a long tube-like structure that plays a role in the exchange of genetic material between bacterial cells. If one bacterial cell has a plasmid that makes it resistant to an antibiotic, it can take that plasmid and send it through a pilus to a neighboring bacterial cell. Through safety in numbers, now it has a friend that's also resistant to the antibiotic, and it's benefiting from that. Fimbriae are proteins that allow the microorganism to stick to surfaces. For example, E. coli have Fimbriae to stick to your intestinal microvilli. Flagellum are tail-like structures that allow for the movement of the microorganism. The glycocalyx is the outermost layer of the cell. It consists of Fimbriae or a capsule. Fimbriae is protein. Capsule is a gel-like layer. The cell wall gives the cell its shape. The cell wall is just underneath the glycocalyx. Bacteria have two types of cell walls, gram-positive and gram-negative cell walls. In a gram-positive microorganism, they have this cell membrane, and then they have a thick layer of peptidoglycan. In gram-negatives, they have an inner membrane, which is a normal phospholipid bilayer, a thin layer of peptidoglycan, and then an outer membrane, which is part phospholipid, part lipopolysaccharide. Peptidoglycan is unique to bacteria, and the thickness and location of the peptidoglycan determines whether it's gram-positive or gram-negative. Gram-negatives are more difficult to treat because they have this outer membrane that provides for an extra layer of protection. Gram-positives are pretty easy to treat because their peptidoglycan is exposed on the surface. You can design any antibiotic to target that peptidoglycan and successfully kill the bacteria. Although many bacteria are developing antibiotic resistance now, particularly MRSA, which is a gram-positive that resists methicillin, which is a type of antibiotic that's usually pretty effective against staph infections. Staining cells distinguishes bacteria based on the cell wall type, so you can stain them in a different way to determine whether they're gram-positive or gram-negative. In lab, we're going to be performing this staining procedure. When you stain the bacterial cell, you can see its cell shape. There's three major shapes that exist. A coccus is spherical, a bacillus is rod-shaped, and spirillum is spiral. There's also these spirochetes, which are really long spirals. There's coccobacillus, which are really short rods, and there's Vibrio, which are bent rods. Some bacteria form thick-walled endospores that survive harsh conditions. Clostridium botulinum, the cause of botulism, is one example. So this is what happens. When this bacteria starts to dry up, it goes into panic mode. It replicates its genetic material and it produces this endospore. This endospore is like a little seed. The whole bacterial cell will then die because the environment dried up, nutrients are limited, and the whole bacterial cell dies. But this endospore stays behind in the environment. This endospore resists basically everything. It can resist UV, chemicals, alcohol, bleach. You can sometimes light it on fire, and this thing will still survive. It does not survive autoclaves, though. So an autoclave is a sterilization chamber. They use that for sterilizing surgical equipment and stuff. We use it to sterilize the media that we use in our microbiology classes. But it doesn't survive that. So it doesn't survive complete sterilization, but it can survive almost everything. And these things can remain for millions of years. As soon as this endospore is planted back in the correct environment where it's surrounded by nutrients, the endospore will hatch open and grow into this big, long bacterial cell. It'll be an actual living bacterial cell. And this endospore could be, hundreds, thousands of years old and all of a sudden come back to life. Proteobacteria form one of the phylum of bacteria. Their metabolic pathways and habitats are diverse. E. coli and Salmonella are types of proteobacteria. Cyanobacteria form another phylum of bacteria. These are autotrophs, and they were the first to release oxygen gas as a byproduct of photosynthesis. So cyanobacteria colonized the earth, or kind of filled the earth with oxygen gas. Other phyla of bacteria include spirochetes, firmicutes, acinobacteria, and chlamydiae.